Welcome to our fourth and final pitch session of the day. We hope you've enjoyed the conference Invest in the West, presented by Amplitude Ventures. It is my pleasure to welcome back Kim Klein, President of Bioassociation of Manitoba, or commonly known as BAM, who's going to introduce and moderate our speakers. Thank you. Over to you, Kim.
Thank you. Thank you, Wendy. Hi there, everyone. Welcome to the fourth and final pitch session of the day. My name is Kim Klein. I'm the president of the Bioscience Association in Manitoba, and we are absolutely thrilled to be able to be here today and be here with you. And I'm really honored to be able to be at this fourth and final pitch session. During this session, we actually have 6 companies that are going to be pitching today with themes of diagnostics, digital health, medical devices, and med technology. So the companies, we have 6 of them today, and I would like to get started right away with our very first pitching is Michael Cross, who is the Chief Innovation Officer of Artemis. Michael.
Production of high value and high quality medical isotopes. And we do this on cyclotrons that are particle accelerators. We are about to initiate a Series B financing of 30 million in order to expand our sales and marketing efforts, acquire a site in the US for isotope production, to continue to develop our platform into other isotopes, including therapeutics, and then for general and admin. Artemis is we feel, a great investment opportunity for a number of reasons. It operates in a rapidly growing market. We have a very well-defined unmet market need. Artemis produces a turnkey solution for the providers of radiopharmaceuticals. And we have our technology already accepted by all of the key stakeholders in that regard. We have an attractive financial return that we feel is very justified. And of course, we have an experienced leadership team. Just as a quick snapshot of our background, we were founded in 2013 from 4 academic institutions that were completing a federally funded research grant to come up with a solution to reactor-based isotope production. And we chose cyclotrons, and we chose to demonstrate technetium-99 as our first isotope for proof of concept. And since that time, we've raised 3 rounds of financing, venture financing, a total of 23 million US. We went from 0 employees in 2018 to over 30 now, and we'll be expanding further as we move into a new site in May. Artemis is built upon peer-reviewed publications and technology awards, which we're very proud of, an innovation and patent strategy around our hardware, consumables, and our processes, and validation through international pharma and biotech collaborations and out-licenses. Our commercialization model is global in nature, and it's based on the razor blade model. For a cyclotron, people that are unaware of a cyclotron is essentially a big bombarder of particles against other substrates. And it was invented by Dr. Lawrence at Berkeley, UC Berkeley in 1930. Triumph has the world's largest one on the top right there. I think you can see a technician in the middle of it. And now they've adopted 2 small cyclotrons at the bottom that are available to hospitals and universities and radiopharmacies. What Artemis does is engineer and produce all of the components necessary to harness the energy that a cyclotron produces and turn that energy into valuable medical isotopes. So we have enrichment of solid targets as one of our key competitive advantages. We make the target capsules that move these targets back and forth between the hot cell, which is in the production suite, and the target station, which is located and attached to the cyclotron. And of course, we have a number of dissolution and purification components. I always describe Artemis in buckets of innovation and giving us our unique competitive advantage. And we have this system hardware of all of the necessary components. We have the solid targets that are cold in nature until they're bombarded by the cyclotron, thereby producing gallium-68, for example, or zirconium-89, copper-64, or the initial isotope that we went after, which was technetium-99. We also then put a lot of effort into processing and understanding how to get that radioactive isotope off of the solid target and into a clean format for the patient. We have a regulatory strategy that is unique and outside of the norms for OEMs that are in this space. And that is that we validate our processes and our stability testing, and we file drug master files based on those drugs. And then we partner with innovators for them to use our radioactive isotopes in their ligands, in their kits. We validate with them. We're included on their INDs and their NDAs, and then give them the ability to reference our DMFs. But unlike other traditional pharmaceutical companies, innovators don't make the drug. They farm that out to a hospital radiopharmacy or a radiopharmacy such as Cardinal or Pharmalogic or Jubilant. And those are the groups that actually do the final production of the isotope and its radio labeling with the kit, and then for preparing for patient dosing. So in that regard, we also have relationships with the radiopharmacies. So we've locked up through our processes and all of our hardware, the innovators and the radiopharmacies that are actually producing the patient doses. Here's a case example, gallium-68. It's a PET imaging isotope that's companion diagnostic to therapies that are coming out. There are currently 2 approved products. One is NetSpot from Novartis, which is for neuroendocrine tumors. And then the other is a PSMA agent by Telix for diagnosing prostate cancer. The gallium currently provided for these 2 radiopharmaceutical kits is through generators. The problem with that is that they only come in very small sizes that provide only 2 or 4 doses of patient doses per day. There's a long lead time and they're quite expensive to acquire. Behind all of this is there are a bunch of other agents in development with Novartis and Isotopia, Point Biopharma. So there's about 10 different products in development, which is moving the 40,000 or so annual scans up over 1 million predicted in the next couple of years. And the problem here is that the generators just cannot provide enough gallium. They do not have the ability to make the generators in the quantity that is going to be required for all of those imaging scans. It's very expensive. Each dose for an imaging scan is about $3,000, and patient care is obviously then going to be impacted, and innovation's impacted as well. But Artemis has a solution through our hardware and our solid targetry, and this has allowed us to demonstrate in a 2-hour production run about 100 times the quantity of activity that a generator produces. And we can do this repeatedly on a daily basis. It's equivalent to the generator. The COGS on a per patient dose are a fraction of what the generators are. And our technology is really compatible with all of the OEM cyclotrons that both installed and that are new. One example of how we can take a market and be able to supply a good portion of the patient doses is demonstrated here by having these relationships with Cardinal, Pharmalogic, Jubilant, who are adopting our technology. And because we produce so much activity, they're able to make that activity, parcel it, and send it out to the areas that are needed for the radio labeling and then the patient dosing. So we can actually ship this activity pretty far and be able to dose the patients required. Over the last 18 months, we've established quite a nice track record of 50 new installations in North America, Europe, and Asia. We produced a world record supply of gallium, over 10 curies. And we filed 3 drug master files to date. We did get approval for technetium-99 by Health Canada, and that was at the end of 2020. And and we'll be filing it with the UK and the FDA this year. We have a pretty strong financial background with the 19 million Series A that was completed in May of 2020, with Deerfield leading that and support from Cork Ventures. And importantly, we have the innovator relationships, Novartis as being one of the key ones. There's been plenty of activity in the space in terms of fundraising, both IPOs, M&As, and our projections are quite attractive when we're selling our systems and the disposables with those systems on a daily basis. It's an interesting space, obviously growing markets, growing investments, lots of M&A. We're going out, as I mentioned, for a $30 million Series B. We have a great base with Deerfield and Cork, and we'll be spending those funds on R&D, the US facility, so that we can make our own isotopes and sell those to the users, and then sales and marketing efforts. Do want to give a big shout out to a lot of financial support that we've received from provincial, national, and international funding partners. And this has been really instrumental in our growth in those early days and even today. And we wouldn't be here without them. So that is a great thing for Artemis. And I'm going to finish off with the management team. Paul Schaefer and I were the co-founders of Artemis back in the early days. And now we've brought on a great team of radiopharmacists and professionals in the space that have a long history of success in radiopharmaceutical commercialization. And that's it. Thank you.
Thank you so much, Michael. You did a great job. I would like to introduce our next presenter, and it is Dr. Anurag Srivastava, and he is with Oncodrex out of Winnipeg, Manitoba. Anurag.
Thank you so much, Kim, for the introduction. Thank you, Life Sciences BC, for the opportunity to showcase our company today. In the last 15 years, we have learned more about cancer than we did in past 100. We have made remarkable advancement in treating cancers, especially breast cancer. Despite that, triple-negative breast cancer remains most difficult to treat, affecting younger women with poor prognosis. At Oncodrex, we are determined to change that. Oncodrex is a clinical stage oncology company focused on developing therapeutics for metastatic cancers, starting with triple-negative breast cancer. I have spent 20 years of my research focused on N-merystoyl transferases. These are proteins that are essential for normal cellular function. This work has led us to some impactful discoveries, which have been patented now. So our discoveries have led to the development of 3 assets in colorectal cancer and breast cancers. In 2012, we discovered NMTs as blood markers for precancerous lesions of colorectum and colorectal cancer. And then we entered into a vigorous research program to understand mechanistic implication of NMTs in pathogenesis of cancer. And that has led to the development of a platform technology by discovering mechanistic pathways that triggers unique molecular signatures for different invasive cancers. So while we have assets in 3 important areas, as a company, we decided to focus on triple-negative breast cancers. We have identified unique molecular signatures associated with NMT, which are drivers of metastases. And we are developing small molecule as drug candidate against unique molecular signatures. And these unique molecular signatures have also allowed us to develop a companion diagnostic that can select responders to our drug candidate. And this is really an attractive and important component for innovative therapeutics program. We are in advanced discussion with two leading CROs, Jubilant Biosys and Evotech, for an integrated drug development program. We have identified and selected a target and have reached the target validation stage. We check the key boxes for innovative therapeutic programs, including triple negative being an unmet medical need, a therapeutic with a companion diagnostic would be a significant differentiator from the current standard of care, except for PD-L1 TNBC. We have intellectual property and freedom to operate. An innovative therapeutic with companion diagnostic to select patients will help us to go for expedited pathway to regulatory approvals. Breast cancer is also a very large market. Triple negative breast cancer therapeutic alone is a $3 billion market. While our strategy is to focus on triple negative breast cancer, but a therapy and also strengthen our therapeutic pipeline, CRC screening test and hormone receptor positive predictive diagnostic are nonetheless valuable assets. So without too much additional time or cost, these two assets can be developed to a point where they will be of interest to strategic buyers. Our CRC screening test is a liquid biopsy test with unparalleled sensitivity and specificity for precancerous lesions, and that sets us apart. For hormone receptor positive predictive diagnostic, it's a simple IHC based test to predict treatment response for endocrine therapy. So we have received over $1 million in non-dilutive government grants and currently have government grants of $300,000 to fund our diagnostic assets. We will be completing our hormone receptor positive validation trial in early 2023 and CRC confirmatory trial in 2023 as well. We are raising a seed round of $6 million to take our TNBC therapeutics to IND enabling stage and build our business team. So achieving each of these inflection points make Oncodrex more valuable as a company. Completing the hormone receptor positive predictive retrospective trial of 1,360 patient would provide sufficient data to make the asset valuable to potential strategic buyers, providing an early exit opportunity. Recent exit in this space is Biotheranostics, which was bought by Hologic for $230 million. Although this company was in a revenue stage, but our diagnostic is much simpler and very cost effective compared to them. For CRC space, it's been a busy space, especially for liquid biopsy. Grail was sold to Illumina for $8 billion, but it's a different company with more than 50 cancers. Whereas Thrive Detect, which was bought by Exact Sciences for $2 billion, is a liquid biopsy company for CRC, which is very similar to ours. Although they were at a much advanced stage when they were bought by Exact Sciences. But our liquid biopsy it has unparalleled sensitivity and specificity for precancerous lesions, and that differentiates us from rest of the crowd. We have a robust intellectual property portfolio, and our team is a world expert in NMT space. We have dedicated full-time scientists, technicians, and clinical staff working in the company, and we have renowned clinicians such as Dr. Narula, Dr. Fang, Dr. Gadi on our team who have already established and sold oncology company in the past. We are raising a $6 million equity seed round to accomplish the milestones we have set out including the potential exit of 2 assets within 24 months. Thank you so much for the time.
Thank you again, Anurag. Well done. Our next presenter is Dr. Max Snyder, and he is presenting the company Primary Peptides.
Thank you very much, Kim, for that generous introduction. It's a pleasure for me to introduce Primary Peptides. We're a clinical stage. Oh my God, we're not that. Claire? You had my slide. Okay, you have my slide again. We're a clinical stage platform technology, pioneering new therapeutics via protein manipulation. As you can guess from our name, Primary Peptides uses peptides as a therapeutic modality. And we think peptides fit really into a sweet spot, both size-wise and modality-wise, between small molecules that are frequently used as therapeutics and monoclonal antibodies. Peptides are just short chains of amino acids, and they have advantages. It's possible to do things with peptides that are difficult to do via the normal routes of drug development. So the next slide shows you the problem that we're trying to solve. And the problem fundamentally is that most drug targets are proteins, but most proteins can't easily be drugged using conventional methods. Our platform technology enables us to hit a lot of these difficult to drug targets using our peptide platform. So among other things, we have the ability to prevent any pair of proteins from coming together. We can block the interaction between any pair of proteins. In addition, we can grab 2 proteins and glue them together using a peptide molecular glue, thus strengthening and stabilizing the interactions between them. In a third interrelated platform, we can grab any arbitrary protein and drag it over to one of the 2 cells' garbage cans for degradation. We're quite different from other companies in the antisense or PROTAC space in the sense that we can degrade proteins in very different ways, and we think with many advantages. We're also high selectivity, low toxicity, we're cell permeable, and our assets can cross the blood-brain barrier. As I mentioned, we're a clinical stage company. We've completed a phase 1 clinical trial that I'll tell you about in a moment, and we have 2 other assets that are in IND enabling studies. We're focused on hard to drug targets in the cardiovascular, oncology, and neurodegeneration space. All of our assets are first-in-class peptide therapeutics. We have signed 3 different drug development partnerships with 3 different companies for 3 different assets, and these are moving along. And we have an excellent patent portfolio with patents issued and applied for in multiple jurisdictions. Next slide shows you a little bit about how our technology works. We're very good at figuring out how 2 proteins contact each other when they bind to each other. And based on that knowledge, we can create various kinds of peptide therapeutics. So on the bottom left, you see a yellow protein and a green protein, and you see the neon green stick figure. That's a peptide which is preventing that yellow protein from binding to the green protein, protein-protein interference. In the middle, you see what I call a 2-handed grabber, which is able to grab the yellow protein with the green part of the peptide. It's able to grab the green protein with the red part of the peptide, and it brings those 2 proteins together, creates induced proximity, and stabilizes and strengthens a protein-protein interaction. And this can be a very powerful therapeutic strategy. On the right, you see a gray circle, which is a cell. Inside it is a target protein. Our drug is the little worm on the outside of the cell body. The red part of that little worm is the cell penetrating domain. It gets us into the cell or through the blood-brain barrier. The protein binding domain grabs that target protein with high selectivity and good affinity. And the third domain in yellow enables us to then traffic that protein to either the proteasome or lysosome for degradation. I don't believe that there's another company that can do things do the things that are described on this slide. The next slide shows you that our technology is more than is more than just vaporware. On the top left, you can see that white area in the mouse brain. That's an area that would normally be destroyed in stroke. But if you look on the right, you see that there is no white area, and that's because of K13, our most advanced asset, which can save over 90% of the brain cells that would otherwise die following a stroke. On the top right, you can see one of our degraders degrading alpha-synuclein, the signature protein inside Lewy bodies in Parkinson's patients. And it degrades it rapidly over 24 hours and does an excellent job. On the bottom left, you can see one of our molecular glues in action, binding a misfolded protein together with its chaperone to be able to reduce protein aggregation to therapeutic effect. The next slide shows you our pipeline. As I said, we're a clinical stage company. K13, our most advanced asset, has completed a successful phase 1 trial in ischemic stroke with robust biomarker and safety data. That's been partnered with a company called Yabo Pharmaceuticals, publicly traded in China. We have 2 other assets partnered with other companies, Simcere and Prime Medicine, in the cardiovascular space. Both of these assets are in IND-enabling studies, and we hope to file INDs and enter the clinic this year. We have a robust pipeline of preclinical assets in the oncology and neuro space, and these are moving rapidly forward toward IND filing, toward IND-enabling experiments. The next slide shows what we're hoping to do with a Series A raise of $25 million over the next 2 to 3 years. We hope to use funds to advance our platform, which is already very powerful and has the potential to become still more powerful with multifunctional peptides, nucleic acid delivery, and more advanced glues coming online. We expect at the end of the 2 or 3 years to advance 2 of our preclinical assets to IND filings and potentially into phase 1, and to advance at least one of our cardiovascular assets through an IND filing into phase 2 in North America. In addition, we hope to expand our team and relocate to new labs and offices. Speaking of teams, the next slide illustrates the wonderful team that we've been able to put together over the last couple of years. All of these individuals are experienced, talented, and really have leading edge capabilities. In addition, we have a blue chip advisory board consisting of individuals with expertise in the various disease areas that we're investigating, in drug development, in drug delivery, and basically in all of the areas that we're trying to work on now. I'd like to thank you. Next slide, please. I'd like to thank you for your attention. We're looking forward to meetings with investors and potential partners, and I'm happy to answer any questions that you might have for me. Thank you.
Thank you so much, Max. That was great. I would like to now introduce our next presenter, Dr. Russell McAllister, and he is going to present to us Bold Therapeutics.
Thank you very much, Kim. Russell McAllister, CEO of Bold Therapeutics, and I wanted to thank Amplitude Ventures and Life Sciences BC for sponsoring this event. Bold Therapeutics is developing Bold 100, which is a revolutionary small molecule therapeutic for the treatment of advanced cancers. Cancer drug resistance is one of the primary limiting factors for current oncology treatments, and Bold 100 works in combination with a wide range of different agents to significantly improve outcomes. Why Bold Therapeutics? It's a very versatile technology. We have clinical data from now 2 clinical studies. We have orphan drug status in gastric and pancreatic cancer with the potential for breakthrough therapy designation later this year. Non-clinically, we've demonstrated consistently and repeated efficacy across a wide range of indications, and we're highly synergistic with other cancer therapies. This is a first-in-class product. It has a unique mechanism of action, minimal competition, and robust global intellectual property protection. And we know what we're doing. This is a veteran team. We've raised more than $1 billion of capital throughout our careers with a number of successful exits. We've negotiated international partnerships, and we have extensive Wall Street IPO and public company experience. Clinically, we have run global clinical trials. We have experience in oncology. We've interacted successfully with FDA, Health Canada, and MFDS. And non-clinically, we've elucidated the mechanism of action, completed 2 cGMP campaigns. We're data-driven with global collaborators, more than 50 academics across the world. I founded the company with Jim Pankovich. I handle the corporate side, and Jim runs clinical development. A corporate snapshot for Bold. We founded the company in 2018, and we've come a long way since then. We just completed our phase 1b study and initiated our phase 2 study, where we're now actively dosing patients. We signed our first licensing deal in 2020, and we expect to sign a second regional licensing deal later this year. And we've done all that on less than $5 million. We're currently raising a $5 million round, of which 3 is already committed. So we're looking for a little bit more Series A1 capital. And in the third quarter, we expect interim phase 2 data, which will drive a $30 million Series B round and 2 additional parallel phase 2 studies. Let's talk about the phase 1b data. In the phase 1b data, we demonstrated that Bold 100 can be safely combined with FOLFOX with no new grade 3 or 4 treatment emergent adverse events. Importantly, patients can also remain on therapy safely for an extended number of treatment cycles. One patient administered 15 treatment cycles, which is very unusual with oncology products. On the efficacy side, the preliminary data suggest a clinically meaningful improvement in progression-free survival over FOLFOX alone in a very difficult to treat and refractory patient population. We even had a patient that had a 48% reduction in target tumor lesions. Let's compare that with Lilly's Cyramza, which was approved in 2015 in combination with the other 5-FU chemotherapy regimen, FOLFIRI. That did about $1 billion of sales in 2021, and you can see the data there. I can't share the specifics of our efficacy data yet since we're under embargo, but I can say it's substantially better than Lilly's Cyramza. Bold 100 has a unique multimodal mechanism of action. It induces cellular stress leading to apoptosis through selective inhibition of GRP78 and ROS induction. As you can see on the right, GRP78 levels are highly correlated with poor patient prognosis. As a result, it's a very attractive cancer target that has not been able to have been dosed successfully previously. Bold 100 significantly improves cancer cell killing. 2 examples here, a solid tumor example with data from Moffitt Cancer Center in pancreatic cancer, and a liquid tumor example, multiple myeloma from Queen's University Belfast. In both examples, adding Bold 100 to standard of care substantially improved outcomes. And these are both highly resistant cancers. It works across a wide range of different therapies. On the panel on the left, you can see we're synergistic or additive with almost every other cancer drug class. And on the right, you can see adding Bold 100 to cisplatin substantially improves mean tumor burden relative to an already sensitive therapy. We have robust worldwide intellectual property with issued and pending patents covering all major commercial territories that don't expire until the late 2030s, so quite long-lived. We executed our first partnership in May 2020 with Hana Pharm, one of the largest companies in South Korea, and we expect to sign another licensing deal late this year. On the capital strategy front, we're currently completing a Series A1 round, $40 million pre-money valuation. We're raising $5 million, which gives us 18 months of runway, and 3 of that is already committed. Later this year, after interim phase 2 data, we expect to raise another $30 million that extends our runway for 24 months and covers the cost for 2 additional phase 2 studies. There's a very significant life-threatening unmet need in colorectal cancer specifically. You can see the 5-year relative survival rates for distant cancer are quite low, less than 15%, and almost all colorectal cancer patients eventually develop metastases. And this is despite the fact that there's a large number of approved therapies in colorectal cancer. They're just not that effective. And you can see in this treatment paradigm that Bold 100 fits very well into the space, covering both non-BRAF and RAS subtype colorectal cancer, as well as BRAF, RAS subtype colorectal cancer. We have a number of upcoming milestones, a non-clinical data presentation at AACR in April, a clinical data presentation at ASCO in June. We expect to close the Series A round concurrently. And then in the second half of the year, we're looking for that interim phase 2 data, efficacy endpoints in advanced GI cancers, closing another regional licensing deal, that institutional Series B round, and initiating additional parallel phase 2 studies. In 2023, we're looking for full data from that phase 2 study and a potential IPO. That's all I had. Thank you very much for the time. And again, thanks to Life Sciences BC for putting on this conference.
Thank you so much, Russell. Great job. I would like to introduce our next speaker, Dr. John Lewis, CEO of Entos Pharmaceuticals. Good luck, John.
Thank you very much, Kim, for the kind introduction, and thanks so much to Life Sciences BC and Amplitude for giving me the opportunity to share a little bit of the story of Entos Pharmaceuticals. So Entos is a clinical stage genetic medicines company based in Edmonton, Alberta, founded in 2017, and we have a pretty bold vision. So as a genetic medicines company, we imagine a world where basically no disease is out of our reach. And with the sequencing of the human genome and the development of some amazing tools like gene editing, RNA medicines, siRNA medicines, really, no disease can be untouched by these. And what we really need to be able to realize this vision are safe, effective, and redosable nucleic acid delivery technologies. Of course, that's the field we're in. As a summary, obviously, there's a genetic component to almost every disease. We've seen with the pandemic, obviously, genetic medicines have come to the fore and their ability to be rapidly developed and even modified to suit changing conditions. But these can be applied, from cancer to infectious disease to rare disease. And the real challenge, though, is that current nucleic acid delivery platforms, either the viral-based ones like adenovirus or adeno-associated virus, or non-viral like LNP, have pretty significant limitations in their safety, biodistribution, and most importantly, redosability. And so we've developed a platform called the Fusogenix proteolipid vehicle platform that we think combines the best features of both the viral delivery platforms and lipid nanoparticles to be able to deliver RNA, DNA, or even protein to any tissue in the body safely, effectively, and repeatedly. Now, we've had a busy couple of years. We have a COVID-19 DNA vaccine that's currently in phase 2 clinical trials. We got some just beautiful data on gene therapy applications, vaccines, oncology applications, and CNS applications for the platform. We've been fortunate in raising significant funding through non-dilutive means, $35 million. And the platform has been validated through 3 recently announced license deals that really only comprise about 10% of our platform, but including a deal we just announced in January, a really staggering deal, over $4 billion with Eli Lilly. We're really excited about that. Now, the opportunity here, we're raising money to take this traction and scale the company quickly to drive multiple clinical programs. And we're hoping that we can find like-minded investors to join us on our quest. We've really assembled just a fantastic team. Founded in 2016 by myself and co-founder, another Canadian scientist, Roy Duncan, who discovered all of the underlying technology. Arun Raturi comes from a great biotechnology and academic background who's really developed the Fusogenix platform from the ground up. Recently been joined by a couple of heavyweights, Steve Chen, with a long background. I couldn't fit in all the logos, but Takeda, Lily, Amelin, and others as Chief Medical Officer. And Jason Ding, really a legendary M&A expert coming from Deloitte Life Sciences in Canada as our Chief Business Officer. We're now a multinational company, again, headquartered in Edmonton, Alberta, now with a corporate office in London, UK, and a brand new R&D facility in San Diego, California. Now, so what are we trying to address? So genetic medicines are really the delivery of tools that either upregulate or downregulate genes or even edit the genes themselves. And so we can deliver these using either viruses or non-viral platforms. Viruses, we've seen this during the pandemic, all of these platforms in use. Adenovirus vaccines of Johnson & Johnson and AstraZeneca. We have approved gene therapies with adenovirus and AAV, but viruses do a really good job of delivering genetic material, to replicate the virus, but they're not the ideal drug. So and they have challenges in manufacturing scale up, their biodistribution, what you can fit inside, emerging toxicity issues in many applications, and of course, because they're viruses, they can't be dosed again and again. Lipid nanoparticles were really developed to address the limitations, and we do have approved systemic therapies and now, of course, widely used vaccines. But for systemic administration, we still have significant toxicity challenges and biodistribution. The toxicity is really linked critically to their mechanism of action, which is what's called endosomal escape. So they get inside cells, they break out of the endosomes, which are inside the cells, but they do this through a biophysical means, and this causes toxicity when there's too much of it in the body. So at Entos, we thought, what if we could combine the best aspects of viral and non-viral approaches? So we do this using a really cool discovery that Roy Duncan made at Dalhousie University some 2 decades ago, where he discovered this class of virus, the only non-envelope virus to make a fusion protein. And this fusion protein is really cool. It's super tiny. It's non-immunogenic, which means we can redose. But when we incorporate it into a lipid-based platform, it allows direct fusion with target cells and delivery of the genetic tools directly into the cytoplasm of cells. Using this protein, Entos has been able to reimagine what an LNP can be and created the proteolipid vehicle platform that we call Fusogenix. And it really, again, combining the best features of viral and non-viral to create a platform that's quite flexible, allowing us to deliver RNA, DNA, proteins, or combinations thereof safely, effectively, and repeatedly. And so we spent a lot of time on creating the platform, working on manufacturing in the past couple of years. We created an extremely well-tolerated platform that's manufactured using a continuous flow, single-step microfluidic process that's scalable. That's immensely scalable. We can make hundreds of million doses of a vaccine, for instance, and we can target these to any tissue. So, this is really the key differentiator slide between PLV and conventional LNP. Because we were able to reimagine the formulation of the LNP into a PLV, it allows us to go after anything in the body. So when we administer, a systemic dose of a DNA gene therapy in a mouse, for instance, at a dose that would kill an animal with an LNP, we get broad biodistribution. So LNPs are typically taken up just by the liver. And you can see here when we dose systemically, comparing a PLV to an LNP, this is at 1 mg and 5 mg per kg, relatively modest doses. You can see highly hemorrhagic livers. This is well above the maximum tolerated dose. Even at 20 mg per kg, plasmid DNA formulation of our PLV completely clean. And this is the reason. LNPs go into the endosome and escape the endosome. All of the immune sensors are in that endosome, toll-like receptors, the gas-sting pathway. So we see these massive spikes, even at this really low dose, 0.5 mg per kg of an LNP, we see a 100,000-fold increase in IL-6. Even at 20 mg per kg, the fast PLV platform is not significantly different than a saline injection. So completely different as far as immune toxicity. So what this means on balance, if we increase the biodistribution to the whole body and we decrease the immune toxicity, we can give systemic doses, and I'm showing data here from non-human primates, where we see liver is the 6th organ down. we get flat biodistribution to all tissues. We can hit muscle, we can hit bone marrow, we can hit stem cells, we can hit heart. So really, and by optimizing the PLV platform, we can optimize it for different tissues. For gene therapy, thinking about single dose and long durability is really important. With a single dose of plasmid DNA PLV given in a mouse, looking 2 months later, we're expressing in a plasmid luciferase, and we can detect it using immunohistochemistry. Basically, every cell of the liver is expressing luciferase, every cell of the lung, every cell of the spleen. So we use this to create a novel gene therapy to build muscle. It could be used in sarcopenia applications and many other age-related disease applications. Follistatin is a muscle-building hormone that is expressed typically by the liver. So we created a liver-specific promoter driving follistatin, administered a single dose to mice, and after 1 year, these mice are jacked. So we see substantial change in body weight, and most importantly, a substantial change in their grip strength. When we look at the muscle, huge increases in the cross-sectional area of their muscle fibers and the muscle weight. So this is a systemically administered gene therapy given once with this pretty amazing outcome. We also have some pretty remarkable data in oncology. We've been able to create programmable suicide gene therapies using plasmid DNA payloads. This particular one, OSX-001, is entering GLP tox, IND-enabling studies. We got really compelling data in immunocompetent models, the CT26 colorectal cancer model. We're seeing complete responses with OSX-001 plus a checkpoint inhibitor. And also in this really difficult B16 metastatic melanoma model, we're basically clearing the lungs of metastatic disease. I mentioned, of course, our vaccine, Fusogenix VAX-001, currently in phase 2 clinical trials. We're getting with a DNA vaccine at similar doses to an RNA vaccine. We're basically getting similar data to RNA, except this is a fridge-stable, single-dose DNA vaccine. So really good antibody titers, neutralizing titers. It's protective in hamsters after challenge, and basically, the lungs are completely clear of viral load after a single dose. So I guess, again, Antos has been busy. We have 2 spin-out companies, Oncocenix and Oncology, developing the oncology assets. I mentioned the OSX-001 gene therapy. We're also working, really excited about in vivo CAR T. Instead of taking the cells out, we're just administering a single therapy to achieve CAR T. Aegis has a bunch of internal programs in vaccines and therapeutics. Of course, we have the COVID-19 DNA vaccine in phase 2, but also multiple other vaccine programs. And again, our previously announced deals with Apellis in inflammatory disease, BioMarin in rare disease, and Eli Lilly, of course, in CNS diseases. So again, I said we're raising money to scale the platform and drive these multiple clinical programs. And please reach out if you're interested. Thank you very much.
Thank you so much, John. Great presentation. I would like to introduce our last presentation today, Dr. Leslie Hill with Mesintel Therapeutics.
Thanks, Kim, for the introduction, and I'm excited to be here today to tell you about Mesintel, where we are decoding fibroblast biology to create superior therapeutics. Mesintel Therapeutics is a pre-seed stage company, and our focus is on fibroblasts, which are a notoriously difficult target with a lot of interest across many different indications. One of the enormous challenges in drug development is identifying robust and effective therapeutic targets, and this is where we come in. Through our proprietary target discovery platform, we're able to identify better targets and ultimately superior therapeutics that address the underlying causes of disease. So fibroblasts are a type of cell found everywhere in your body, and they play an important role in every aspect of health and disease. They're the decision makers and organizers, and they lead the way for other cells. So you can think of them as being similar to an orchestra conductor. Fibroblast dysfunction leads to disease, including but not limited to some of the ones shown here, such as cancer, aging, and fibrosis. The widespread and diverse roles of fibroblasts make them an excellent therapeutic target for a number of conditions with high unmet medical need and enormous market opportunities. So at Mesintel, we focus on decoding or better understanding fibroblast biology, and we're taking a unique approach to achieve this. In comparison to other companies and groups that focus on a single tissue or disease, we follow fibroblasts in any and every context. At the core of the company is big data. We've used cutting-edge technologies to comprehensively map fibroblasts, profiling their biology in more than 15 tissues and numerous pathologies, including multiple cancers and fibrosis. And this is what truly sets us apart. No one has this much data on fibroblasts. Despite enormous efforts, many questions remain on how to treat these diseases, and we believe the answers lie in our fibroblast-centric data. We utilize our proprietary data and analysis platform to identify high-quality disease-related targets. To better understand the underlying mechanisms of action and to evaluate these targets, we validate our data in a number of different ways. These include working closely with clinicians to assess targets in human samples, as well as incorporating private and publicly available human datasets into our analyses. We evaluate our targets of interest in vivo in a variety of human biology-informed genetic murine models and through the use of a variety of in vitro assays, using primary human and mouse cells. These innovative, comprehensive, and complementary strategies allow us to really understand the biology and mechanisms of these targets, accelerating their validation and therapeutic development. Using our platform, we actively mine this data to better understand the pathways and programs that underlie fibroblast dysfunction. There are many ways that fibroblasts can behave dysfunctionally, including through alterations in proliferation and matrix production, as well as increased or decreased factor production, a consequence of which, for example, is altered immune responses. By defining the mechanisms responsible for a specific dysfunctional state, we're able to identify disease-driving targets for therapeutic intervention. As an example of the utility and the power of our platform, I want to quickly discuss our lead program, soft tissue cancers or sarcomas. These types of tumors are classically defined as being immune cold. Notably, a high percentage of patients progress to advanced disease, which is associated with a poor prognosis. At Mesintel, we have used a novel humanized mouse model to determine the key events that lead to cancer progression. One of the major highlights is that we're able to follow fibroblasts from the pre-cancer stage all the way through to full-fledged tumor development. And this is really unique. No one else has a sarcoma model that allows you to study this whole process. Other models only capture one end of the spectrum and none of the intermediate stages. So utilizing cutting-edge single-cell omics technologies, we've acquired a dataset of over 100,000 individual cells collected at various time points during sarcoma development. And using our platform, we have identified key inflection points and determined new mechanisms responsible for fibroblast dysfunction and the immune cold sarcoma environment that allows these tumors to evade the immune system. Interestingly, many of the pathways, programs, and targets that have been identified in sarcoma are also active in the tumor microenvironment, and this is another area of research that we're actively pursuing. Ultimately, we utilize our wealth of data in health and in disease to decode fibroblast dysfunction and identify tractable targets that can be modified with therapeutics to positively affect a number of diseases. Our lead programs are sarcoma and the tumor microenvironment. We are actively investigating a number of other indications. Since there are many commonalities between fibroblasts and different tissues and diseases, targets for one disease are likely to translate to others. We are planning to begin seed round fundraising once we have secured the terms of our license with the University of British Columbia, which we anticipate to be finalized by late spring. To push our pipeline forward, we plan to use these funds to support business development efforts, formalize our scientific advisory board, to further develop our platform, to complete sarcoma IND enabling studies, as well as to further progress other indications to achieve proof of concept data. In addition, we will pursue non-dilutive funding through multiple sources in order to maximize the investment impact. Our business model is based around our target discovery platform that provides us with the opportunity to develop multiple therapeutics and partnerships. This will be achieved through in-house development and commercialization, out-licensing, as well as platform partnerships. These avenues will drive platform validation and enhancement, as well as funding. The Mesintel team contains individuals who have held multiple roles in academia, biotech, and pharmaceutical companies, and who collectively have over 60 years of research experience. For us, fibroblasts are a core part of the team, and we look forward to further discussing how we have recruited fibroblasts to work for and not against us in treating disease. Thank you for your time and attention, and we look forward to hearing from you and further discussing our company. Thanks.
Thank you so much, Leslie. Great job. I just want to say many thanks to everyone and congratulations to our presenters today. Michael, Anurag, Russell, John, Leslie, and Max. Those were great pitches. Each of you did such an amazing job. I'm always excited to see and hear about the innovation, the research, and the hard work that's happening here in Canada and Western Canada especially. Please know that I wish each of you much success in your endeavors and future accomplishments, and of course, with your investments. Well, this was the final pitch session of the day. I want to thank all attendees for joining us here today. We hope you enjoyed and learned something new. Please don't go far because coming up next, we have a very interesting panel discussion entitled The Power of Convergence, Exploring the Intersection of Biology and Technology, which will start right after a very short break. Take care, everyone, and see you there.